Good afternoon and welcome to the Center for Vaccine Preventable Diseases seminar series. And today we're also have the pleasure of co-hosting this seminar with the McGill Epidemiology and Epidemiology, Biostatistics, Occupational Health at the School of Population and Global Health and Faculty of Medicine at McGill University. I'm Sheza Fadl. I'm an assistant professor in the Clinical Public Health Division at the Dalai Lama School of Public Health, and I work at the Center for Vaccine Preventable Diseases. Throughout this year's series, we will feature researchers whose works align with the CVPD's themes. Those are vaccine hesitancy, value and impact of vaccines, immunization ethics and governance, and immunity and community protection. We are thrilled to be co-hosting today's sessions with McGill University. And in today's session, we are also very looking forward and thrilled to have Dr. Senjuti Saha, who will be presenting on tackling pediatric infections in low resource settings. After Dr. Saha's presentations, I'm also really happy that Dr. Nicole Basta will be also co-moderating this session, and she'll be facilitating the Q&A sessions after Dr. Saha's talk. So before we start, I would like to acknowledge the traditional territories of the Masaga of the New Credit First Nation, Anishinaabe, Wendat, Huron, and Haudenosaunee indigenous people on which the Dalai Lama School of Public Health now stands. The territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and the Confederacy of the Ojibwa and allied nations to peaceably share and care for the resources around the Great Lakes. We'd also like to pay our respects to all our ancestors and to our present elders. McGill University is also situated in the traditional territory of the Kehenka, a place which long served as site of meeting and exchange among nations. We recognize and respect the Kehenka as the traditional custodians of lands and waters of which we meet today. So I will start by also introducing both Dr. Basta and Dr. Saha. So Dr. Saha, our primary speaker, is a scientist at the Child Health Research Foundation in Bangladesh, working at the intersection of molecular biology and public health. She is an alumni of University of Toronto. After her completing her PhD in molecular genetics at U of T, she moved back to Bangladesh to work on the front lines in public health. Currently, she focuses on pediatric preventable infectious diseases with the goals of using modern molecular technologies, including on-site metagenomics to identify etiologies that evade standard laboratory testing, establish genomic surveillance to track and understand molecular basis of antimicrobial resistance, and estimating indirect impacts of vaccines on the overall health system of resource-constrained settings. Her work is grounded in advancing health and research equity in Bangladesh and beyond. She is a member of the Polio Transition Independent Monitoring Board of the Global Polio Eradication Initiative and an associate editor of the BMJ Global Health. I'd also like to introduce Dr. Nicole Basta, who will be moderating the session. Dr. Basta is an infectious disease epidemiologist and vaccinologist with experience in designing and implementing epidemiological studies that generate critical biological, clinical, and behavioral evidence. The goal of her research is to optimize disease prevention and control. She draws on her background in epidemiology, biostatistics, ecology, and evolutionary public health and global health to advance our understanding of epidemiology of multiple globally important diseases. So I'm going to go ahead oh yeah, before we go in the housekeeping, the other additional housekeeping is to remind that after the talk, there will be Q&A sessions. Feel free to raise your hand function so that Dr. Basta will call on you, but also feel free to put in your question in the chat. And without further ado, thank you, Dr. Saha, and I'm looking forward to talking to you.
Thank you for that introduction. I'm so excited to be here. And also very sad that I could have taken this opportunity to come to Canada, but it didn't happen. But hopefully I'll impress you guys so much that you have to call me back next year and I can. visit Canada in person. I am going to share my screen. Yeah. Okay. Okay. So as Shaza was already saying, I'm going to talk about how do we work with pediatric infections in low resource settings. And I was really debating on what I should present. I mainly focus on meningitis, and for a long time I was like, that's the talk that I'm most uncomfortable giving. Maybe I'll just talk about meningitis. But I also thought that perhaps it's more important to talk about some of the basic biology work that we do in Bangladesh and how difficult it is to work in Bangladesh. But all of that can have impact on what vaccines do we introduce, which vaccine is going to have the greatest impact. And at the same time, I'm giving this talk in the midst of COVID, and cases in Bangladesh are spiking very, very quickly. So it also seemed unfair not to talk a little bit about COVID. So I'm going to touch upon both typhoid and COVID a little bit. Okay. So this is a slide that I start with most of my talks, because this is really what motivates me to do my everyday work and motivates my entire team at the Child Health Research Foundation. Over here, what I'm showing you on the y-axis is the number of cases, infections caused by Haemophilus influenzae type B. That's a bacteria that causes meningitis and sepsis. This is the number of cases we admit per year in Dhaka Shishu Hospital, DSH. That's the largest pediatric hospital of the country, also where I work. So these are the number of cases we were seeing every year. And then in 2009, the vaccine was introduced. And right after the vaccine introduced, it's a fantastic, phenomenal vaccine. Cases dropped down very, very quickly. Right now, this vaccine is estimated to prevent about 3,100 deaths of under 1 children every year. And this is deaths, not cases. And this is, again, a great news. However, this comes on the backdrop of the fact that this vaccine was introduced 21 years after it was licensed in the US for use. So, 21 years, we did not save those lives that we could have. And those are the gaps that we are trying to close constantly in our team. So I work for the Child Health Research Foundation. It's a not-for-profit and an NGO in Bangladesh. And our mission is to improve child health in Bangladesh and use that data to perhaps also improve health around our country in South Asia, and use whatever we find in our lab to advocate for changes in policy decisions. So what do? We focus primarily on neurological and respiratory pathogens. So, pathogens that cause meningitis or pneumonia. We also do fever investigation. So fever of known or unknown origin, like typhoid, paratyphoid, or any kind of neonatal sepsis, for example. And at the same time, we are based in the largest pediatric hospital. So oftentimes we have to switch gears, even if we don't want to. For example, in 2017, we had the first and the largest chikungunya outbreak. In 2019, we had the worst dengue outbreak ever since 2000. So we immediately had to switch all our gears to then tackle all of those diseases, or figure out what tests to do, what are the data we are going to generate. And we use all of this data that we generate through our surveillance and laboratory work to influence policy decisions on vaccines, and sometimes also diagnostics. And actually work by this foundation facilitated introduction of the hip vaccine in 2009 that I already talked about, and then the pneumococcal vaccine in 2015. So as I already said, for today's talk, I'll talk a little bit about typhoid, about antimicrobial resistance, and how very basic experiments in the lab can actually influence policies if we really know what question to answer. And then I'll go on and talk about how the capacity that we built in order to understand typhoid better, how that capacity came in handy when COVID hit Bangladesh. So let me start with Salmonella Typhi, the cause of typhoid. It's a human-restricted gram-negative bacteria. and it is transmitted by contaminated food and water. And typhoid is a huge burden. There are about 14.3 million cases per year, 135,000 deaths per year, and it's a neglected tropical disease. And I have the neglected in bold and in quotation because it's so neglected that for decades it didn't even make the WHO neglected tropical diseases list. That's why we work on it. So there have been 2 vaccines that have been also available for decades. Recently, there have been 2 additional vaccines. Those are conjugate vaccines that can work also on very young children. So it can be prevented by vaccines, but it is because it is transmitted by contaminated food and water, any kind of improvement in water, sanitation, and hygiene will also prevent typhoid. So I already said that there are about 14.3 million cases per year. Where are these cases? According to a GPG paper that was published in Lancet, most of the burden exists in South Asia. So mainly India and Bangladesh, and also Pakistan bears a huge burden. And it is estimated that 500 to 700 per 100,000 people get typhoid in Bangladesh. Now, if you ask me, I know that number is really high, right? But if you ask me, I think that's an underestimate. Why? Because I got typhoid. My brother got typhoid. All my cousins got typhoid. If you go into my lab meeting and ask people to raise their hands of who got their typhoid, I kid you not, 90% of the lab is going to raise their hand. Because it's like the rite of passage to become a Bangladeshi. We have to get typhoid. So we think it is an underestimate, and it is, because most of this work that is published is through modeling. A very recent study that was actually conducted in South Asia, it was a prospective surveillance that was conducted in Bangladesh, India, Bangladesh, Pakistan, and Nepal, showed that indeed the burden is high. Bangladesh has the highest incidence in the world of about 1,260 per 100,000 cases per year. And we often joke, should we report how many people get typhoid, or should we report how many people don't get typhoid? The good thing about typhoid, though, is it can be treated by antibiotics, and that's why mortality rate is actually quite low. So in the pre-antibiotic era, in the early 1900s, 20% of the kids, at least, who got typhoid died or had severe complications. But with antibiotics, mortality has come down to less than 1. And the first antibiotic that worked for typhoid was actually Preventable, that was introduced to treat typhoid in 1948. And because antibiotics work overall so well for typhoid, it also means most of these children, or most of the patients, can actually be treated in the community, can be treated in the outpatient department, right? Because they can just be given oral drugs. Like I wasn't admitted, my cousins, my brother, none of us were admitted in hospital. We got a blood culture done, or someone told us we have typhoid. My dad went my mom went into the pharmacy, got us antibiotics, we got better. But the problem with Typhi, like with many other organisms in the world right now, is we are seeing an increase in antibiotic resistance. So, Typhi has shown resistance individually to every antibiotic or antimicrobial that has been used so far. And I guess the epitome of that is a recent outbreak that is happening as we speak in Pakistan. So in 2016, an outbreak started in Pakistan by a strain of Typhi, by a strain of Salmonella Typhi called extensively drug resistant, or XDR. It is resistant to all the classes of antimicrobials listed over here. That outbreak started in 2016 when, our collaborators work in Pakistan, and we were closely monitoring it, and there were only a few cases when the outbreak started. But as we are monitoring the outbreak, we're seeing that the proportion of all the typhoid cases that are extensively drug resistant are basically increasing. So that strain, it's a clonal strain that's extensively drug resistant, is taking over the entire Salmonella Typhi population. And we're very worried because there is only one oral drug that now works against this XDR Salmonella Typhi. And that drug is azithromycin. If XDR typhi becomes azithromycin resistant, we won't be able to treat these children in the outpatient departments anymore. So this is very, very concerning. Now, when the outbreak was first discussed in a typhoid meeting, I was in that meeting. Of course, we were very worried. But there was this idea that perhaps Salmonella typhi doesn't become resistant to azithromycin, because azithromycin is a really efficient drug, it's cheap, and it's also, some people call it a wonder drug, because there are some trials that have shown that if you just massively give azithromycin to kids, apparently it decreases mortality. So there was this idea in the scientific community that Salmonella typhi cannot get resistant to azithromycin because we haven't really seen that much resistance. But then on the 13th of April, one fine morning, I was reading the New York Times, and there was our very good collaborator, Eric Mintz of CDC USA, actually said what we were all worried about. And he said that it's a matter of time. It will become resistant to azithromycin. It's just about when it becomes resistant, and are we prepared by then? And that really struck me. That struck me personally, because I started thinking, why do we not know more about this? Why isn't anybody working on azithromycin resistance? So I had just started working at the Child Health Research Foundation. I immediately went into our database and I tried to figure out how many strains of Salmonella typhi do we have. It turns out that between 2009 and 2016, we had about at least 939 typhi strains that were just stored in our -80. So immediately, my team pulled them out of -80 and tested them all for azithromycin resistance, because that's not a regular test that we do. And out of those 939, 12 strains actually ended up being resistant to azithromycin. And the resistance started emerging in 2013. Azithromycin came into the market of Bangladesh around 2004, but its use really took off around 2008. So it kind of made sense that resistance started appearing in 2013. And azithromycin is good because it's like one dose per day. So people really like having that antibiotic, and it's cheaper. Okay, so then I started digging into the literature, because I didn't know much about typhi at that point. I was just learning about this bacteria. So I, dug really deep, and I'm trying to understand what causes resistance, And at that point, absolutely nothing was known about what causes resistance of azithromycin, against azithromycin in typhi. So then what can we do about it? How can we generate that knowledge sitting in Bangladesh? Well, we took those 12 strains and we actually had to ship them out of the country to get them sequenced. We did not have the facility to sequence. We shipped them out and then we got the sequences back, and some of us in the team have a bioinformatics background. So we started looking for what are the antibiotic resistant markers, because from the genome sequences, we should be able to tell what is guiding resistance. And there are this really easy to use online tools where you just put on the genome, upload the genome, it tells you what are the resistance markers. Not a single program out there could tell us what is the resistant marker. That kind of told us that perhaps it is something novel. So then if it's something novel, we have to figure out what is that novelty. So at that point, another student was doing a PhD in our lab, and for his PhD project, he had sequenced another 462 Salmonella strains, typhi strains that were sensitive to azithromycin. So then we were like, okay, that's cool. Now we can just manually compare them all and find out, is there any mutation, anything that is unique to these resistant strains? So this is a phylogenetic tree where basically the most closely related strains will cluster together. And over here, what I've done is colored in red the strains that were resistant. What was really interesting to see, and later you will realize that it was really lucky, is all of these resistant strains clustered together. That means they were very similar to each other, which also means it should be easier to find what is causing resistance. So if I zoom into this further, we were comparing resistant to sensitive. I just picked a few sensitive strains that are most closely related. And you can see all the resistant strains are clustering together. These are the sensitive strains. This is the genotype that they belonged to. And then we looked at all of their resistance profile in the lab, so phenotypic resistance. And we see that all of their resistance profiles are identical to all the antibiotics. Again, another proof that they are actually related to each other. Then when we kept on looking for, what mutations are unique in these 12 resistant strains, we found three unique mutations shown over here, 3 red lines that were actually in genes. One gene had no hypothetical function. One just seemed like an enzyme that was probably not relevant. But what really caught our eye is that this gene over here called ACRB. And I'll tell you in a minute what is ACRB. So ACRB, the gene is pictured over here. And when we looked closely at this mutation, we could see that because of that mutation and there was a change in the amino acid, and R became a Q, and this was in the 717th position. And I'm defining this because we'll keep on coming back to this. So this mutation at the 717th position is called R717Q. Why were we interested in ACRB? Because this gene, ACRB, codes for a protein that is part of an efflux pump. What's an efflux pump? An efflux pump is basically a pump that sits on the membrane of bacteria and pumps out different molecules, including antibiotics. So what happens is the antibiotic tries to enter. So this is the outside of the bacteria. This is the inside of the bacteria, the cytoplasm. The antibiotic tries to enter, but this pump doesn't let it enter, just pumps it out. So whatever this mutation is doing is basically causing this pump to work better and pump out the antibiotic before it can actually kill the cell. So it kind of made sense. So we came up with a hypothesis, and our hypothesis is, but this is all theoretical, right? We're sitting on a computer in Bangladesh and coming up with this hypothesis that a mutation in ACRB, R717, causes azithromycin resistance. All theoretical. Obviously, any scientist, any bench scientist would criticize me and say, but how do you know? Like, where's your empirical evidence? Did you introduce this mutation in a sensitive strain and ask if it becomes resistant? So, I would have to do this experiment in the lab. But there was no way I could have done all of those experiments in Bangladesh because there were many barriers. I wouldn't get like the reagents in time. It would take me months to just set all of that up in Bangladesh. So I was in Bangladesh. My husband lived in Canada, and I was supposed to come to Canada for Christmas that year. And I told him, you know what? I won't come to Canada. Let's take a vacation in San Francisco. Because in San Francisco was my graduate school friend who just opened a lab, and turned out his student was going away to get married. So he had a little bench free in his lab. So I called him, his name is Joe Pontitanemi. I called him, I was like, hey man, I really need that bench for a week. I will bring everything, just let me do the work. I'll just use your reagents. So he said, you cannot work with Typhi in my lab. I don't have the permission, it's level 2. However, you can work with E. coli. So he ordered everything I needed. And my husband's name is Yogesh Huda, and that was our first. He's also a scientist. That was our first project together, the best Christmas ever. We went to San Francisco and started working. And we knew that we had 1 week to do all of this in E. coli. We had to work 24/7, and another week we would have to travel to Stanford, because in Stanford I had another colleague who had the experience of who had the permission to work with Salmonella Typhi. So we knew we would be backpacking a little bit. So first we did our experiments in Joe's lab in UCSF. And what I'm showing you over here is that if I take a simple laboratory strain of E. coli that has either no ACRB gene or has an ACRB gene that has no mutation, then the minimum inhibitory concentration of azithromycin is really low. However, the moment I introduce this mutation, minimum inhibitory concentration rises by 8-folds, telling me that indeed it is that mutation that is making this bacteria resistant. This was all done in E. coli UCSF. Over the weekend, we traveled to Stanford, booked another Airbnb, and did our Salmonella Typhi experiment, and our results were reproducible. Super exciting. We needed to publish immediately because we knew this has huge public health impact because there's this outbreak ongoing. A vaccine was just pre-qualified by WHO, and countries are trying to decide where to introduce the vaccine. And if we could tell the policymakers, look, XDR strains are just one mutation away from becoming ban resistant, perhaps that would guide a little bit of policy decision to immediately introduce the vaccines. So we submitted the paper. The reviewer immediately got back to us saying, it seems like a one-off thing. The work was done only in Bangladesh, and there is no validation from any other country. So we don't, while we think it's a really good experiment, we don't think this mutation is ever going to take off. Okay, no problem. We knew this had to get published. Also, this was all unfunded work because it was vacation. We submitted to PLOS NTD, immediately got accepted, and we also didn't have to pay anything, which is amazing. But what does this mean, right? Okay, so now we know that just 1 point mutation, again to reiterate, 1 point mutation can make a strain azithromycin resistant. 1 point mutation is really easy to acquire. And if XDR strains take up this mutation, then it will become resistant to all oral drugs. That means our outpatient departments who are treating, or the community clinics who are treating typhoid patients cannot treat them anymore. They will all need to be admitted in the hospital. There is no way Pakistan's health system or our health system can deal with that, because 90% of all typhoid kids are actually treated in the outpatient, which means if this happens, mortality will increase. We continued working. So, we immediately disseminated the data. We presented this in the typhoid conference. It was very well accepted. And then we did further work. And we showed this is not a one-off thing. When this resistance emerged, it was just, you remember those strains that were really, really closely related? But as people are using more and more azithromycin, so that data was till 2016, and we continued doing the work, and we now showed that this mutation is just showing up in different strains of Salmonella Typhi. That means it's spontaneous, it's random. Nobody knows which strain is going to acquire that mutation. Again, that means XDR is getting close, can acquire this mutation anytime. So this was a little easier to publish. We followed up again, and we showed bioinformatically that we only worked with Salmonella Typhi, but if we looked at every Salmonella, whether Typhi or any other serovar of Salmonella, this mutation is very common in the strains that have already been sequenced by different other groups and uploaded. It's just that nobody ever looked at this mutation. So then we came up with a recommendation that perhaps rethink mass azithromycin administration. Because while we all want a silver bullet to decrease mortality, this is probably not the best way of decreasing mortality. But we also know that people, it's really important. We know that it's really important that we track this mutation. But the people with the highest burden of Salmonella Typhi or typhoid also don't have genome sequencers to constantly sequence and find this mutation. So then my team actually invested a lot of time to figure out the low-cost approach. It's a simple PCR. It's called mismatch amplification mutation assay, where the PCR is designed such that if there is a mutation and you do a PCR and run a gel, you get no band. And if there is no mutation, you get a band. It costs less than $2. We did that, and we also showed that it gives the exact same results as whole genome sequencing. If someone wanted to use this technique to track this mutation, for example, Pakistan. And it's been a great experience so far. You know, those AMR online platforms who could not tell us what caused azithromycin resistance now can. They have incorporated our finding into all their platforms. This mutation is not a one-off thing anymore. Several other scientists have found it all around the world after us. At least 10 other countries have reported it. And genomic surveillance has actually been put in place to look at this mutation specifically. And this work that we have done, along with some of the other projects that we were doing at the CHRF, actually got us a grant to get our own sequencing machine. And this is why I mentioned in the beginning that we had to ship out our samples. We don't have to ship out our samples anymore. Something, and I want to write to that editor who wrote me that when we first submitted that paper, but I'm going to take the high road. On February 12th, actually, the CDC came up with an alert that extensively drug-resistant salmonella is also spreading in the US, and they are not related to travel. So unfortunately, it's very scary. But what we did in the process is, because it was like questions that we are trying to answer, because we're trying to guide policies on typhoid vaccines. And these are questions that we are very excited to answer. This really ended up empowering, educating, and engaging local scientists at CHRF. There's several people involved. We learned cloning, we learned transformations, how to look for clones of bacteria. They learned a lot, and these are the people who are the front lines of public health. These are the people who have probably had typhoid in their life. And since then, we have done a lot more sequencing. Now we sequence different kinds of viruses, different bacteria. We are doing the cousin of typhi, paratyphi. We're looking at paratyphi now. However, just as everything was taking off, COVID-19 hit. And I feel like all of this work that we were doing before COVID-19 was kind of a practice to deal with something like COVID-19. Like, we were building capacity because we knew subconsciously maybe something like this is going to happen. So Bangladesh detected, I'm going to now switch from typhoid to COVID-19. Bangladesh detected its first 3 cases of COVID-19 on March 8th. And on March 24th, because we are one of the only labs who actually does a lot of pediatric surveillance, we are a WHO Sentinel site specifically for vaccine preventable diseases. So government of Bangladesh contacted us and asked us to serve as a COVID-19 diagnostic lab. It turns out the same day the Gates Foundation asked us if we would be willing to serve as a diagnostic lab, provided they gave us the funding, so everything worked out. We spent the next 5 days between 24th and 29th preparing to become a testing lab. It's a complete switch, right? We work on pediatric. COVID-19 affects all ages and all populations. And of course, it's a virus not only us. At that point, nobody had worked with in the entire world. So there was so much to figure out. But in 5 days, we got everything ready. We used our experience. By 29th, we started testing. And by 31st, we even had detected the first positive case in our testing samples. The lab, the entire team was extremely scared, but they were also really excited because we say we do public health. And it was like we were actually doing public health in real time. We were serving the country, serving the population, scared. We don't know what's going to happen. At that time, nobody knew how it spreads really. Do you wear a mask? Do you not wear a mask? All those questions. But we started to, we got ready and we were excited and we were ready to handle hundreds of samples per day. But we were only receiving 10 to 20 samples. Till we started testing, everybody was complaining. Nobody's doing tests, nobody's testing. Where do we give our samples? There aren't enough tests. And now we start testing, there are no samples. So what do? Right? We had to ask, what are the barriers in receiving samples? And we figured out that there were actually 3 barriers. One is there is overall confusion and within people of where and how to give samples. There's a lot of stigma. At least in Bangladesh, there was a lot of stigma. Landlords were kicking out their tenants if they tested positive. Doctors were not allowed to go back to their apartments. Actually, a lot of my team couldn't go back to their homes because they were testing in the lab. So what we did is we actually have a group of community health workers who work in the various typhoid projects, trying to understand how typhoid spreads. And we can immediately use that group of amazing community health workers to become this phone line people. I don't know what they're called. Like we gave them like 6 cell phones. We distributed the number publicly, and anybody could call us. We could answer their questions. And as that helped a little bit, and what we also realized is we were testing and collecting some samples, but most of the people who were collecting the samples were in these small community clinics. And they had no experience of collecting samples. They had no experience of wearing a PPE or how to dispose of it. So we built a training program. Again, using all the experience that we have had in the past years. We built a training program. We did online training, offline training on how to collect specimens and how to send them to us, kind of like a train the trainer program. And finally, I'll say the most surprising problem was the quality of samples. So these are some pictures of the samples we are getting. Over here are some tubes. These are actually not sticks or swabs. These are sticks from coconut trees that they picked out. And they just put some cotton on them and put them, we don't know in what liquid, and send the samples to us. This is actually good compared to this. This is a Ziploc bag with 2 tubes with cotton stoppers. By the time the tubes reached the lab for us to test, there were more liquid in these bags than in the tubes. So we realized that, okay, we also then need to figure out how to ensure that people are collecting good samples. So we actually designed a kit where we would distribute this kit that contained collection media, a swab, instructions in Bangla, a form, because a lot of these samples were had no names or no phone numbers, so we had no way of communicating with them. So we actually designed all of this and we started distributing them so that they could give us quality samples, and our work was actually valuable. And I said there was a lot of stigma. A lot of people couldn't go back home. And also this was during lockdowns. We did not know if they go back home, will their families be safe? Because in our culture, we live with different generations, right? With parents, grandparents. So we actually set up accommodation in every bit of free space that there was. This was a little gym that we had. It's gone. People sleep here. Now it's used for something else, but that gym's never coming back. We call them the COVID-19 rescue team. We also built a dashboard so that we could be anywhere, because we knew we couldn't be physically close. And every sample that came into the lab was barcoded. Anybody sitting anywhere could track every sample. We knew the epidemiology, which age group is most affected, and what sample are getting, how many are positive, etc. Where are they coming from, etc. And as all of that is happening, remember, we built that sequencing capacity because we were interested in understanding typhoid. That came in handy. So very quickly, as COVID-19 started spreading, vaccines were going into clinical trial. And from previous experience, we know that strains or variants circulating in different places can actually, vaccines can have different impacts on them. We have had that experience with the pneumococcal vaccine. We have that experience with the meningococcal vaccine, and we didn't want that to happen again. So we used that sequencing machine that we got to sequence the first virus in Bangladesh. So all our previous work actually came in handy, and the first sequence was uploaded on May 11th. This is the sequencing team. Of course, it's a pre-COVID-19 picture. That's why there are no masks. Since we sequenced, several other groups started sequencing, and hundreds and hundreds of samples have already been sequenced. And we don't even match up to them. We have sequenced about 150 samples, but I think our proudest moment is that other people realized that we can do this in country. We don't have to ship out our samples, because we are so used to just shipping out samples that we don't even realize what our power is. But just one group showing, look, this can be done in Bangladesh, kind of spread this confidence that everybody started working on it. What we do now is actually look at more the bioinformatics platform, because now sequencing is getting easier and easier, but handling that much data can be difficult. So we actually host the main next train build for Bangladesh. We have an amazing bioinformatics team that does it. If you want to look at the evolution of SARS-CoV-2 in Bangladesh, just hop onto this website. We'll get an updated view of variants, evolution, etc. And then, as all of this is happening, in the midst of COVID-19, we graduated. We started with a small sequencer that I showed you. We graduated to a bigger one. So now we can sequence a lot more. Now we can do hundreds and hundreds of Salmonella Typhi actually in our lab and do genomic surveillance. And what we are doing at the moment is actually building this pediatric disease observatory in the country, because we've been doing surveillance. That's where, all the Salmonella Typhi strains came from. So, we have Salmonella Typhi, we have like all the gram negatives, for example, that cause neonatal sepsis. But how do we take that simple epidemiological surveillance into a 21st century surveillance where genomics and on-site surveillance are connected? So we're building this comprehensive observatory where bacteria are whole genome sequenced, viruses are whole genome sequenced, and also if there are cases of like unknown origin, like meningitis or fever, how can we use RNA metagenomics to diagnose them? And that's our next phase, is do genomics, do 21st century surveillance, but very, very comprehensively. Of course, we have a lot of catching up to do. These are just some of the strains that we have been very carefully leaving in that -80 from where the Salmonella Typhi came. If Salmonella Typhi has a story, trust me, every species, every serovar of bacteria has a story. We just need to go and dig them out and hopefully not have to go to San Francisco again. So yeah, we are working towards establishing real-time genomic surveillance, not only for bacteria, but also viruses. And I think I'm out of time, so I'm going to end over here. I'm very privileged that I got to come here and give this talk and meet all, the wonderful people here. But it's all teamwork. Every person in our team has a role to play. Everyone makes us who we are. It's a huge team. This is just a part of the team. We have about 350 people. Not only our team in Bangladesh is amazing and very hardworking, we also have an amazing group of collaborators all around the world. And we are very grateful for all the support we get. With that, thank you.
Excellent. Thank you so much, Dr. Saha. This was a really fantastic presentation, and we really appreciate that you're sharing your expertise with us. It's amazing how quickly you were able to pivot, and it's wonderful to see the discoveries that you and your team have made so far. I can't wait to hear more about the surveillance system that you're setting up for the future. If anyone has any questions, please enter those into the chat. We already have a couple of questions queued up, so I'll begin with our first one. Teresa Yorkas is noting that this is very exciting work, and she thanks you. And she wanted to know, how does your lab and hospital interface with government labs and government hospital systems? And how does your data enter into the COVID public health recommendations for the population, or COVID surveillance?
Excellent. So let me start with COVID, and that will probably give a picture of how everything else works. So COVID, when we became a COVID diagnostic lab, we became an extension of the government. And so basically we were acting, although we are an NGO, we were acting as a government lab. So every specimen that came into our lab, it had to be registered with the central government system. Every report, we didn't give reports to patients directly. Every report was actually sent to the central, kind of the CDC Bangladesh, and they're the ones who distributed the reports. And those reports were basically text messages. All the data goes into the government repository, and then they have their committee to decide on vaccine introduction. We have a little more role. So our executive director of the NGO actually also sits in the committee who decides on which vaccine to introduce or where and what are the phases. So we can also get the have a little more influence based on our findings. So this is COVID. For a lot of the other typical surveillance that we do, we are a WHO lab. We share all of our data. And of course, because we work with the hospital has all the data, and anything that is unique. For example, once we found a chikungunya meningitis outbreak, that's absolutely unknown. So the government wouldn't even ask for it, for example. So we'll report, right? We'll let the hospital know, do a little press release, so people are aware of what is going on.
Excellent, that's really helpful, thank you. Our next question comes from Ankur Sudin, who notes, great work, thank you for this presentation. And the question is, what policy decisions have you been able to advocate for using this amazing surveillance results that your team has been capturing in Bangladesh?
Oh, my favorite question. So, I am a new member at this NGO. I only joined in 2016. Before that, on and off, I volunteered or interned. So it was actually our data that introduced the Haemophilus influenzae type B vaccine in 2009. So before 2009, there was actually this idea that Hib disease did not even exist in South Asia because nobody was looking for it. So it was, again, that executive director who now sits in those committees. He built the microbiology laboratory in that hospital, the first microbiology laboratory, and then he started looking for Hib. And once he started looking for Hib, he generated surveillance data, and that really is what facilitated the Hib vaccine in the country. And then nobody else works on Hib in the country. And then there's the pneumococcal data. Again, there's an entire team dedicated to working with Streptococcus pneumoniae or pneumococcus. What are the serotypes? And based on that, our group actually not only worked with the government in Bangladesh, but was also went to the British Parliament to get funding to get the vaccine, and worked with Gavi to introduce the vaccine in 2015, PCV10. And now, not only did we help introducing the vaccine, we are also looking at the impact of the vaccine on the disease.
That's excellent. I think I see a lot of students and trainees here today, and I think that, that the true impact that you've had is going to be really an inspiration to those who are entering this field. So thank you for sharing that. Our next question is from Geber Menden, Geber Tekel, and apologies if I've mispronounced your name, who says, great work and very interesting talk. How do you see the overall impact of COVID-19 on antimicrobial resistance?
Oh, it's horrible. It's bad. It's really bad. Our antibiotic consumption really is off the charts in the country. Anybody has a little bit of cough or a sore throat, they will buy all the antibiotics that there are, because anybody can buy antibiotics over the counter in Bangladesh. There are prescriptions for COVID-19 going around on Facebook. Facebook is really popular. And those prescriptions consist of like many, many different vitamins and also antibiotics that you should be having for 8 days. And of course, one of the antibiotics is azithromycin. So I think we are heading towards a very, very big crisis.
Yeah, your work on this area is really incredibly timely, especially with the studies that have indicated that the widespread azithromycin use could have some impacts, but the implications for antimicrobial resistance is really important to consider as the trade-offs. So thank you. Our next question is from Deeman Roy, and Deeman says, thank you for this interesting talk. Bangladesh is recently experiencing a growth in infection rate. So do you think Bangladesh is going to experience a variant behind the surge for COVID-19?
I didn't understand the question. So Bangladesh is experiencing a very high rate of infection at the moment.
Yeah, and I believe the question is, do you think that is going to be due to an as yet undefined variant that's driving that surge in COVID-19 cases? Or do you think there are other factors at work? That's my interpretation.
I mean, what we know from COVID-19 is, yes, variants are playing a role. There's no doubt. There's scientific evidence that now shows that some of the variants transmit better, right? But at the end of the day, it's about human behavior. It's about, are you following health guidelines? Are you wearing the mask? Because no matter what variant we have in the country, the precautions at the moment is going to be the same. So the spike that we are seeing right now, definitely there will be some mutations. We are already, from our genomic surveillance, we're definitely finding variants. We're finding the UK variant, which is known to transmit at a higher rate, like it has increased transmission rates. We also are finding the South African variant, which can escape immunity. So there are definitely problems, but at the end of the day, it is our behavior.
Yes, the more we can do to reduce transmission, the less of a risk some of these.
Yeah, I mean, we're giving bodies for the virus to mutate. If we don't give bodies to the virus to mutate, they will not mutate.
Mm-hmm. Very nicely said. Thank you. Our next question comes from Ankur Sudan again. With indiscriminate use of antimicrobials for COVID-19, including those approved by country governments, what new antimicrobial resistant issues do you anticipate seeing or have you seen in your surveillance data, I believe with your new genomic surveillance program?
Yeah, so I think the time span of COVID-19 and use of antimicrobials isn't long enough for us to see that impact yet. In the bacterial population, number one. Number two, remember, we work with a pediatric population, right? And it's not the pediatric population who is directly exposed to the antibiotics, it's usually the adult population. So it's going to be even later by the time we see it, see the resistance in the bacteria that's causing infection in the pediatric population. But I suspect that it's going to be much higher. The rates of antimicrobial resistant infections is going to be higher.
Thank you. And our last comment so far is from Therese Boucher, who says, this is really outstanding, Dr. Saha. She says, I'm overwhelmed with admiration for your reactivity, competence-based creativity, and commitment to this work. And I think that is a sentiment all of us are feeling right now. So thank you. Please, if anyone has any other questions, enter them in the chat or feel free to raise your hand. And while others are queuing up, I think I'll ask a question of my own, if that's all right. And so, Dr. Saha, you have really highlighted a number of really interesting challenges, but also opportunities that you've taken advantage of in your work with the Child Health Research Foundation. I wonder if you could speak a little bit more about what some of the ones are on the horizons, the challenges and opportunities that you see, and what you hope will change in the future to help overcome the challenges and enhance the opportunities a little bit better.
Right. Let me start with the opportunities. The opportunities are how a little bit of work can go such a long way. I worked in Canada. I lived in Canada for 11 years. I did my PhD over there. And the work I did was very, very, very exciting. But I had a really hard time by the end of my PhD seeing how that work is going to benefit. And that was on antimicrobial resistance also, where a lot of my experience comes from that. It's very hard for me to see how that work would directly impact the population of Bangladesh. Because there is so much, they're so behind. For example, I will not see drug development from my PhD work that will actually be implemented in Bangladesh in my lifetime, probably. So I think in Bangladesh, the opportunities, the infection load is very high. So there is more opportunity to work to prevent those infections, and so much is unknown. There are so many low-hanging fruits, and I work in the field of microbiology. Infections are my livelihood, so definitely that's an opportunity for me. Although I hope infections go away one day and I lose my job. Failures. Not failures, or I guess barriers, kind of. What are the challenges that we see? Challenges are getting, for example, supplies and regions. It's not like I couldn't have done any of those experiments in Bangladesh when we was working on Salmonella Typhi. I would have to order all the enzymes for cloning, all the primers for the PCR, etc. But sometimes it can take months for supplies to come into the country. And we have no funding to do that work. It also costs a lot more to buy actually reagents in Bangladesh, which is very counterintuitive, because a lot of these companies that make these reagents and supplies are based in rich countries, and they don't have companies or local branches in the country. So we have like this middleman or distributor who also makes a large scale of profit. So we end up paying a lot more. And there's of course customs and tax. It becomes really expensive and it's time consuming. And we have been scooped before just because we were waiting for one simple reagent. And another thing I will say is very difficult for us is access to literature. And this is something I took for granted when I was in Canada, because I could read any paper I wanted to, because there was this U of T library, and McGill I'm sure also has a library where you just read whatever paper you want. And then I came to Bangladesh and that just stopped. So I don't have access to library anymore. So very often students or young scientists will come and be like, I really wanted to present this paper, but I can't because I couldn't access the paper. So the paywalls are very difficult for us, like thanks to Sci-Hub, but not all papers are even in Sci-Hub, but Sci-Hub is amazing. And also to publish, like for example, this work that we did on azithromycin, to publish it in so-called elite high-impact journal would be like $3,000 to $5,000, even with the tickets to San Francisco. I don't know how much I actually spent on those experiments to be able to pay that much for publication. So those are definitely some of the barriers that are very difficult for us. And people don't trust us. I don't know why. And that's also a very, very demoralizing barrier I think we face constantly.
Thank you so much. That's a great range of insight that I think is going to help us figure out what some of these challenges are that we can overcome in the future to make sure that there are more opportunities than challenges in this type of work. We've got a number of excellent questions here in the chat. I think since we're running a little short on time, I'm going to summarize one or two of them. So the next question is about the idea of appropriate technologies, and some areas of research believe that there's a need to adapt appropriate technologies to low and middle income settings. But in your work, you've really thrown that idea out the window and shown that you can use the technologies that you have at your disposal to try and answer questions that are locally relevant. And so the question here is, what would you say to those who are advocating for this appropriate technology mindset, the need to kind of adapt approaches used in high income countries for low and middle income countries?
Oh. I mean, it's not like, I don't think it's a black and white answer, right? It's not like, we cannot accept any of the technology or we should be accepting every new technology. It's about how do we use whatever we have, but also get better, improve. But somehow we have to get out of that vicious cycle of, no technology, no data, so no technology, right? Or no we are stuck in this vicious cycle. So somehow we have to break it. And when you're initially trying to break it, you have to optimally use whatever you have. At the same time, I have used a lot of the technology from outside, like sequencing is nothing that I had at my disposal. It is something that I had to learn from my friends in high-income countries. I work very closely for who helped me set up the sequencing capacity and work with them constantly. But at the same time, I also don't believe that everything needs to be an innovation. Everything needs to be like a disruptive healthcare or disruptive innovation. I mean, there is a lot of innovation and I don't think we use them enough. I think what I preach and I really believe is it's not about constantly making innovations, it's about whatever is there, how do we take that innovation to where it is needed the most. Right now, Bangladesh needs the innovation, South Asia needs the innovation, Sub-Saharan Africa needs the innovation, but are these innovations reaching them? Even like that's something that happened 10 years ago, even have those reached these regions? I think those are the questions we need to ask ourselves.
That's excellent. Thank you so much. I see we're just running up against the end of our session. So I apologize to those whose questions we didn't get into, but I want to be respectful of Dr. Saha's time. As you can see, she has a lot that she needs to work on and we need her to continue working on all of these exciting areas. So please join me in thanking her for this really wonderful talk. We're really pleased that you joined us today, and hopefully we will have you back to visit in Canada at some time soon for a real vacation. It looks like you need one after all of this effort. So I will pass the floor back over to Shaza for closing remarks.
Excellent. Thank you. I think on behalf of everyone here, I think many of us are completely floored and about your experience and your innovation and the way that you've dealt with some of the barriers in building capacity in an equitable way. And I think the comments keep being added to the chat too as well. Thank you, everyone. There's an evaluation link. As I remind everyone after these seminars, please do take a couple of minutes to evaluate. These are always important for particularly early career researchers to collect those and include those in their work. And so this is just take a couple of minutes to evaluate the award. And we look forward to everyone joining us for our next seminar. So thank you very much, everyone, and have a good rest of the day.